Hello. Hello, can you hear me? Great. So, good afternoon, good early morning, whoever, where are you? So, I want to give an introduction to ChemRICH, um, chemical set enrichment analysis software for metabolomics that we developed a couple of years back with Oliver and teams at West Coast Metabolomics Center. So I'm now leading my own lab at Econ School of Medicine at Mount Sinai, New York. And the lab is working on developing integrated data science solutions for metabolomics and exposomics. And there were ideas around improving ChemRICH to include compounds that were kind of missed in the first version of ChemRICH. So I updated the software and wanted to give an introduction to those updates in this webinar. So before we dive into set analysis, ChemRICH, and why we need to do it, we all need to remember that metabolites are not independent. Levels of a metabolite can depend on levels of other metabolite. And we know that from these biochemical maps or metabolic network maps that they are interconnected, that changes in a one pathway or one reactions can have a short distance or long distance effect on other metabolites level. So concept here is that metabolites are not independent. So any computational approach we develop or build, we have to keep this concept in mind that metabolites are not independent. Why set analysis? Now, before we go to ChemRICH, I need to explain that why we do a set analysis for metabolomics. So I took a hypothetical example scenario. In a traditional approach, so in a traditional analysis, you have, let's say we measure these 10 metabolites, metabolite 1 to 10, and they, let's say, belong to two sets, set 1 and 2. These sets could be anything, class, pathway, modules. And we have these p-values. So p-value here, and red color p-value and blue color p-value. So, and red color p-values are actually significant that they passed 0.05 cutoff, and blue color p-values are not significant that they did not pass 0.05 cutoff of p-value. Now, a traditional approach is that we apply a multiple hypothesis testing and we correct these p-values, and we get a second set of p-values where metabolite 3 from 6 were considered that they were not significant because their p-value did not pass a second criteria after correcting for uh, multiple hypothesis testing. And if it is not true, meaning that after correcting for multiple hypothesis testing, we converted those significant metabolites that were truly significant into non-significant ones, so the red to blue, that means it was a type second error, it was a false negative. And that also means that we might have missed a biological insight that they were significant, but they were turned into non-significant because we were assuming that each metabolite is a single hypothesis, which is not the case. Because if they belong to a set, that means metabolite 1 to 6 are from one set, therefore it is one hypothesis, not 6. So we are basically applying extra penalties or unnecessarily penalties on individual metabolite level, whereas the analysis should really happen on the set level, that the actual hypothesis if the set is significant or not individual metabolite. And that's why we need to do set analysis, so we do not increase the type second error, which is false negative, and meaning that we do not miss any new biological insights from your data sets. Now, untargeted metabolomics, this is really important because untargeted metabolomics is a discovery tool. In many settings, it's a discovery tool. So in that case, we are looking for new biological insights. So it's something we never studied or we completely missed. And applying this multiple hypothesis testing on individual metabolite p-value, we will be missing those biological insight because this p-value correction is causing an increase in false negative rates. Now, that justify that we need to do set analysis. We need to find what are the metabolite sets out there and what definitions we can use for sets. So here's a simple table of 4 well-known metabolite sets. So the top row is a pathway map where we say that these 4 metabolites belong to this one pathway, for example TCA, and the pros of this approach is that these are well-known definitions accepted for 100 years or more, and there are canonical maps and they're easy to interpret. The problem with these pathway map definition is the boundaries are manual, that they varies from one database to other. The coverage is poor, that not every detected signal in a metabolomics data set is mapped to a pathway map. Then we have chemical classes, like you take a group chemically similar compounds and we give them a class name, for example sugars or lipids or phospholipids. Now these are accepted by many epidemiologists. There is a good coverage. Almost every chemical compound with a chemical structure can assign a chemical class, and they're non-overlapping, so you could have distinct chemical classes. I don't foresee any problem with or disadvantage of using chemical classes, except that the interpretation could be a little tricky sometimes. Then we have network modules. These are based on reaction networks, that compounds that are mapped to a biochemical reaction, whether experimentally observed or in silico reactions. If they are mapped into a network graph, we can use module detection methods for that graph and detect these reaction network modules. Then the correlation module is, instead of using a reaction as a link between two metabolites, we use a correlation between two metabolites. and correlation could be computed based using metabolite levels in a metabolite data set. So as you can see that from top 3 rows, you will need to have a good idea about a chemical structure, and the last one where you can work with known and unknown both compound, because correlation doesn't need correlation computation do not need knowledge of a chemical structure. You can compute that on both type of compounds. Now if we look at a typical metabolomics data dictionary, the annotated, unannotated compounds can be divided into these 6 pillars. So the bottom one is a majority of peaks have no annotations. Like we don't know what, we only know M over Z and retention time, but we don't know what the substructure or what's the class or what's the other IDs are for these majority of peaks. Then we have substructure annotation based on mass spectra, that take a mass spectra and we compute a substructure based on the fragmentation pattern, but we only know a substructure, we don't know the exact structure. Then we have named but no structure. So this is a class level annotation we get, for example, in lipidomics assays, where we get triacylglycerol with double bond count and carbon chain count. So there is a class and some idea about carbon chain and double bonds, but we don't know the exact structure of acyl chains and the location of acyl chains. Then we have structures where we know the exact structures, like a Smiles code or Inchi, but we don't have a KEG ID, which means they were not covered in a biochemical database. Then we have compounds with KEG IDs, that KEG or a biochemical database have a literature supported evidence that this compound was detected or measured in a particular sample. Then we have this top small fraction of compound that are linked to a biochemical pathway map. So that is only a small fraction, the top blocks here, that only a small fraction of compounds are linked to pathway map, and majority of others have either known structure or a name or a substructure or no names. And pathway maps works only for the top blocks. ChemRICH 1.0, which is the previous version, that works with compounds having Smiles code, so top 3 blocks. It will not work with named but no structure, substructure, or peak with no annotations. So that inspired us to update ChemRICH, that why not be more inclusive, and we create several set definitions that will allow us to include all these different type of chemicals and annotation levels. So one issue was which statistical test to use for set analysis. And hypergeometric test is traditionally used. It's all hypergeometric or Fisher exact test is traditionally used. It was adopted from genomics, where genetic variants or SNP data were passed through a p-value cutoff, because SNP mutations are actually random and they are independent from each other, so it makes sense there. And we also know that in genomics the total number of gene is known, which is n. In metabolomics we have no idea what n is, like how many metabolites we can have in blood or in a normal E. coli cell or any other tissue type. This n is not known. So if n is not defined, we cannot use hypergeometric test, because just by playing with the size of database, so here we see in this plot, just by playing with the size of database, that you go from 20,000 to 100,000 compounds, you can reduce your p-value just by playing with the database size. So hypergeometric tests really depend on a background database, which do not exist for metabolomics matrices or metabolomics assay. Each analysis gives you the different set of chemicals that varies from one lab to other lab to third one. Even within the lab, we will see that analysis done 1 year ago and analysis done today will have a different set of chemicals because the libraries were improved, because the methods were improved. So any statistical methods for a set analysis that depends on this N, a background database, is inappropriate, and it's using wrong assumptions. So ChemRICH 1.0 was using chemical structures that you need to have SMILES code, and it basically takes the SMILES code, it predicts mass classes, which are medical subject headings from National Library of Medicine, NIH. It's a really large ontology. Almost 130,000 chemicals have been annotated with mass classes. So ChemRICH takes a chemical structure, it links it to mass class, and then apply a Kolmogorov-Smirnov test, KS test, which is a p-value distribution based test that it doesn't need a background database, it doesn't need a p-value cutoff, it takes all the p-values for a particular set. For example, in this case, let's say unsaturated triacylglycerols, all the p-values for this particular set were utilized, and they were compared against a p-value distribution under null hypothesis, which will be a uniform distribution, and then you get a second set level p-value here. So here unsaturated TZ, you get a set level p-value that actually can be adjusted further for multiple hypothesis testing, because we do get like 70 to 80 metabolite sets for today's metabolomics data sets. Now that was ChemRICH 1.0, and we know that it only works with SMILES code, peaks having SMILES code. So, and it's hard to include other peaks that do not have SMILES code. So ChemRICH 2.2 is developed, 2.0 is developed, and what we did was, what my lab did is that we kept the original 1.0 top 3 blocks that we can compute the mass classes. Then we can add user-defined sets to include the chemicals having names but no structures. So let the user define chemical classes for them, or user can define a class level SMILES code. Then substructure and class level for spectra and chemicals where we only have a class level information. Then to include peaks with no annotation, correlation modules, and mass spectral similarity modules. So with these six approaches, we can basically include every detected peak into a set and use those sets for conducting set analysis. So that way we do not miss any signal from an untargeted metabolomics data set. So now I'll walk you through the website and what are the options available on the websites. So the website is cambridge.idsl.me, and IDSL stands for Integrated Data Science Laboratory for Metabolomics and Exposomics. So let's go to the website. Okay. So the address is cambridge.idsl.me, and I would say it's straightforward. Follow, but there could be a little rearrangement on going from the top to bottom, because I think I'll put some of these tabs under here. So if you're using ChemRICH and you want to run on your local machine, you need to install it. It's an R package. It's not a package yet. It's a bunch of scripts right now, and I will bundle them into our package soon. But you can actually use the scripts directly by running this code here. The requirements are that you need the latest R, which is either 4.0 or the latest version. You need RStudio, and you need to have latest Java installed and configured correctly, because ChemRICH need RCDK package to compute substructures. I tested only on Windows, but I think it should work on Linux and other operating system, because these are really standard R packages. So once you run this block of code, it will basically install a lot of R packages. It might take a couple of minutes on your computer to install it. And these packages are used by different ChemRICH scripts. So, and that's why it's a good idea to install all of them together. So when you run it next time, you have all the package that ChemRICH need already installed in your laptop or desktop. Once you have all these packages installed, and hopefully you won't see any error, because these are standard packages, and I'm sure you won't see any error. If you see error about Java and RJava, there are solutions on internet that how do you fix the RJava errors. So input file for ChemRICH is always an Excel sheet, and the minimum input you will need is shown here, that you will need compound names. This could be any compound names, and if you have a duplicate, it's okay. You need an order. So order decide how the minimum input files ChemRICH analysis x-axis will look. So because as you can see that in this table we only take order, p-value, effect size, and set. So these sets will be organized by the order. So amino acid will come after amides on the x-axis, and p-value can come from student t-test or any statistical test you do or regression models you run. Effect size is either a fold change, which could be a ratio of median or mean between two conditions, or the beta coefficient. We have a script inbuilt that detects if there is a negative sign in the effect size. It will figure out that if it is a negative association or positive association, or if all the values are above 0, it will figure out that it's a fold change computation. Now this is easy. This is an easy input compared to previous ChemRICH, because previous ChemRICH will need that you have to have Smiles code and PubChem and Inchi keys, which are hard to get for many compounds, especially lipids. It's really tedious work to get those database identifier for lipid molecules. But we simplify it this way, that a user can define their own set classes, and they can run ChemRICH using this minimum input. Now I won't read it. These are the explanation of what I just said, that what the compound name is, what p-value, effect size, and set is. To get the compound identifiers, you can click on get compound identifier. It is using PubChem compound identifier exchange service web page directly, so you can actually copy and paste chemical names here like synonyms, and you type a chemical name, anything you want, like glucose or whatever the list of names you have, and you can select CID or any of the IDs you want. If you want CAG IDs, click on registry ID and select for CAG should be here, GHIJK, yeah, CAG here. So they will give you, PubChem will find CAG IDs for the input chemical name or whatever input we have, CID, Inchi key, Smiles. It will convert them into CAG IDs. And PubChem services using the most updated data in the PubChem database. So we always get the most corrected or most comprehensive identifier exchange. Now, the first was the minimum, where a user have flexibility to provide chemical classes, and it's a free flow input. They can define chemical class, they can define disease, they can define any way they can group metabolites or chemicals. Now, many times we have structures, but we don't have the chemical class information, and so in that case we recycle the code from old ChemRICH, that we can use the chemical structures to predict mass classes. So to predict mass classes, you need three input: compound name, PubChem ID, and smiles code. And what the code will do, it will add these three extra column: cluster number, x log p, and mass class. So x log p is calculated by RCDK using the smiles code. Cluster number is a chemical similarity cluster, and a mass class is assigned either by lookup, that if the compound is already covered in the mass database, or computed based on the similarity. So you will have this column added. Now, I wanted to warn that since these are predicted mass classes, and there's always some error, you will see. So it's always a better idea to go through these mass classes, and I would say about 5 to 10% could be a little off, but not too off, because we are using a higher similarity threshold. So some of them could be a little off, and you may want to talk to a biochemistry expert who knows these chemicals, that does this mass class make sense for this particular chemical. And that way you can actually create your a minimum input for running ChemRICH, that once you get the chemical classes, you can use the x log p to define the order and run ChemRICH. Now, next thing is predicting correlation modules, which to include all the compounds. And so idea here is that we take input MS1 intensity data, then we pass it through a dynamic tree cut algorithm that defines modules based on how the correlation tree looks. And it's similar to WCGNA, weighted network analysis, but we are adopting the idea that those correlation modules can be used as set definitions. So if you do not have example data, here I provided a link to these example data on each ChemRICH sub-analysis, that you will find links. Example data for correlation module is here, which looks like you have first column compound name and then intensity values for all the samples. So these are different intensity values. In this example data set, we have 878 compounds, and many of them are unknowns. As you can see, this starting with X, some numbers, these are all unknown metabolites. So when we run the correlation module analysis, I highlighted this particular set, where we can see that these are all dipeptides, and these three unknowns are correlating with these dipeptides, which pretty much indicates that these unknowns are actually dipeptides too, because they're part of this larger set. And this is a powerful way to rank unknowns. So if you have a mix of known and unknown metabolite together, if you run a correlation module analysis, and if you find these unknown metabolite correlating with other known metabolite in a set, most probably they have some similarity in the classes, and that way you can rank based on the Y value, that we want to look at these unknown first because they were most significant. They were part of the most significant sets, and they're also similar to other chemicals. They are similar, like dipeptide in this case. So most probably these are dipeptide. Somebody just need to look at their MSMS spectra and figured out that what the amino acid composition of these dipeptides. Now, as you can see that we have many clusters, and that way it become a powerful approach. In my opinion, it's a really powerful approach that how we can prioritize or rank unknown metabolites in different sets. For each analysis, there is a script. Yeah, I forgot to mention that but what it's doing, that it's downloading the script from GitHub, then it's loading all the packages that you already have in your computer. Then it runs a function and using the input Excel file. So you can basically download this file and update the data with your data and save the file in a folder. For example, here correlation module detection folder, and then run the scripts. And it will give you a PowerPoint and Excel and a web application where you can actually mouse over and get a detail about different metabolites in each set. So that's the correlation module that includes almost every signal. So these are different ways ChemRICH can be run. So I explained the any set definition, that you have flexibility. So for example, this is the example file for any set. I explained starting a couple of minutes ago that you have a compound name, order, p-value, effect site, and set. The set definition could be anything. So it could be a pathway, it could be a class, it could be disease or a phenotype, a species, taxonomy. You're free to explore how you group metabolites and run ChemRICH to see if that set definition gives you any statistically significant set. And the procedure is simple, that you download the example file, you update the data, you save the data in new that Excel file in a new folder somewhere on a computer, and run the script, and it will generate these 4 files, like a diagram, a PowerPoint, Excel, and an interactive web page, which you can interact to see the composition of different sets. So in this example, we're using pathway sets, which is known overlapping pathway sets from a data set by Metabolome. Metabolome is actually unique in this sense that their tables contain non-overlapping pathway definition sets. So chemical classes, so input for chemical class is similar to the one we saw previously, but you need to have SMILES code in it. So no PubChem ID, no INCHIC ID. We're simplifying that as long as you have a SMILES code, and you have to have a chemical class that you can actually get from mass production script. So if you have a SMILES code, you can predict the mass classes and then combine them both together and use the input file for ChemRICH for chemical classes. And well, what it gives you, that it gives you on x-axis it organizes the classes by x log p. So you have polar compound classes on the left side and non-polar on the right side. So you can see that tocopherol and unsaturated fatty acids, and these are non-polar guys. They are on right side of the this plot. And polar compound like oligosaccharides or sugar acids or inositols, they are on the left side. So it gives you a nice view of chemical sets organized by their lipophilicity. Now lipids are tricky, as we know that they have a different annotation schemes or naming schemes. For example, lipid species could be PC48.5, and that could be either that or that, so we don't know the structure. And if you don't know the chain position, then it will have an underscore that it could be 18.0 or 20.5. And that makes it hard to get a Smiles code, because to get a correct Smiles code, you need this type of annotation that we know the acyl chain position, and we know what the length of acyl chain or what the double bond count. Now, since we know that ChemRICH works on a set level, so even if you don't know this information, we can use a representative chemical that represent a phosphatidylcholine class. So we can adopt ChemRICH in that way that, for example, I got this data from a study on metabolomics workbench. I don't remember the exact ID. So here we say that these are deoxy-DHC, the different chemicals, but they all can have a same Smiles code representing this class deoxy-DHC. And then we can keep going and we can have class specific. So this coenzymes, and you have one Smiles code representing them. LPI, we have one Smiles code representing that, all the LPI. So this way, ChemRICH can actually compute x log p using the Smiles code, or substructure using this Smiles code, to organize these lipid classes by lipophilicity on the x-axis. So in this study, study is actually coming from this liver sample from old versus young mouse. It has 35 classes. So 35 classes, and that means we only need to collect 35 Smiles code. We don't need to collect Smiles code for every single measured lipids. And then it organizes that we have more non-polar lipids like cardiolipins on the right side, and polar lipids like LPS or LPI on the left side of this plot. And the y-axis is how significant these different lipid molecules were in a old versus young mouse comparison. So it's a liver lipidome in old versus young mouse. So we can see that TGs were really low altogether in a liver in an older mouse compared to a younger mouse. Now the next is for unknown, and the subclass and substructure code is still under development because of we need to figure out that how do we handle the MSMS spectra, and how do we compute these classes by different approaches. So most probably we will use NIST hybrid search to compute these classes and use those classes as a substructure or subclass for set analysis. But I think I will do a separate seminar just on this, that how do we compute subclasses for spectra and using them in the ChemRICH. So correlation module, I explained previously that how do we compute it. Here we took a data from cord blood samples from birth cohorts, and in this study so all the compounds were without names, like there was no annotations, but we still could see almost 189 co-regulatory modules. So we can detect that many correlation modules, and then 77 were significantly associated with birth weight, FTR of 0.05. And these cluster 93 and cluster 77 had the strongest positive association with birth weight. Now these are still clusters of unknown compounds, but we can see that it provides a nicer way to rank these unknowns, that if we want to know more about that what chemicals are associated with weight that should go in a structural elucidation or further MSMS experiment, then we can think about that, yeah, it should be cluster 14 and cluster 77 and 93 first, because they were positively associated, and they were most significant positively associated clusters. So that way, if you have a matrix with no identity, like all the unknown features or peaks, you can use a correlation module detection to define their sets, and then you can prioritize unknown. Now still, these unknown need further experiments, that what is the similarity of all the features in this cluster 93, that why they correlate, and are they belong to the similar chemical class, or are they belong to similar pathway, or are they belong to similar origin, and so on. These are the questions that we could now ask to this the ranked set matrix. So if you have metabolomes data, so metabolomes data have, where is my input? So metabolomes data comes with two columns. They call it sub-pathway and super pathway. These are non-overlapping pathway definitions. And the data that I use came from this paper. In the supplementary section, the data table is available. And actually, this is here. Yeah. So this is that we use the Smiles code and the pathway definition provided by Metabolome. And we get a plot like this one, where we can see that if you use pathway sets used by or provided by Metabolome, the sub pathway one, not the super pathway one. And then the second plot we can create is take the Smiles code and compute the mass classes, and then see that if mass provides us different chemical classes that were missed by the metabolomes ontology. We could see that there should be some distinction, because metabolomes ontology is driven by pathway and mass is mostly driven by chemical classification. So they could be complementary, that mass can highlight metabolite sets or set definitions that provide a different angle of interpretation compared to a pathway angle. So my suggestion is that if you have metabolomes data, you want to use the sub-pathway as a set definition in ChemRICH, but at the same time, if you have Smiles code for those chemicals, or even if you get the lipid data, then Smiles code for class level, Smiles code for lipids, you can use those to compute mass classes and compute the lipid ChemRICH analysis. And that way you can generate 2 or 3 views to look at metabolomes data, and they give you ideas that which angle the data can be interpreted. And we will get the similarity, like here we get dipeptides in both ontology, the metabolomes ontology and the mass ontology, because it's a chemical class. but we don't see many of these pathways in the mass ontology and many of the chemical classes in the metabolome ontology, vice versa. So they're kind of complementary. Now, often we have more than one classes compared. So, for example, if you run a mouse knockout experiment and you compare 5 or 6 knockouts to the control group, and so you have to run 5 times ChemRICH, and so there was a feature request for many that they don't want to repeat ChemRICH one by one for 20 comparison or 100 comparison. It would be nice to have a batch script. So now we have that, and the input, oh, actually, yeah, this one. So input is pretty straightforward: name, and you need a class column where you define a class. It could be mass class predicted, user defined class, correlation module, anything you like. But it can work right now only with Smiles code. So you have to have a Smiles code because you need to use that for x-axis. We will add another script there where we can include all the unknowns, meaning that the dependency on Smiles code will not be there, and we can include compounds not having Smiles code in this MultiCambridge script. And to provide the statistical result, you need to have condition A p-value fold change, condition B p-value fold change, condition C p-value p-value fold change, and so on. So you can have condition C, D, and so on. But for each condition, you need to have a pair of p-value and fold change. And ChemRICH will detect that these two columns belongs to condition A, these two columns belongs to condition B, and it will then repeat analysis number of time the conditions are in the input table. So that, and since this is the MultiCambridge script is evolving, we will add also the regression models results into it, and also unknown metabolite, as I mentioned a moment ago. And it's nice to see multi-Cambridge analysis, because then you can actually see into this animation type of view that how different chemical clusters or sets are changing from condition 1 to 2 to 3 to 4, and that helps in ranking the sets, that for interpretation or to highlight most important results, we want to focus on this set first, and then this set, and then this set. Okay, so that's all for today. I wanted to give an overview that we have updated ChemRICH scripts. Eventually, will be turned into a new R package. And now, if you don't want to, there is a question: how do we get the Smiles code for your compound? If you click on Get Compound Identifier, oh, well, okay. I think the PubChem service is down. So using that PubChem service, you can get the Smiles code for from chemical names, from CIDs, from KEG IDs, or any other IDs. It should be up soon. So this PubChem Identifier Exchange service is the way to get Smiles code for your input data. If you don't want to install ChemRICH and you just want to prepare a input file and submit input file to our server. So I have set up a server where you can actually use any of these templates, and then you can submit that Excel file, and we will compute the ChemRICH analysis for your input file using our servers. And the good thing about that is we will always have the most updated code on our side, and different other databases or other uh, approaches that are in development that will be available only on our servers. So um, if you want to use any of these 7 templates in ChemRICH analysis, we have our scripts available on our GitHub, but you can also create the input Excel file, upload to your Google Drive, and then you need to share that link and submit the link here for that Excel file, and select the analysis type and submit, and our server will compute the analysis and send you an email with the result where you can download the result. So this way, if you're having trouble with installing ChemRICH on your computer, which I hope it won't, these are pretty standard R packages, so you should be able to install and download. But in case you're having an issue with R, or you couldn't install or couldn't run it, you can use this forum to submit the ChemRICH analysis, and our server will send you an email with the results. So there is a question that the correlation module is based on what? It's only based on the intensity values. So, and the question about MSMS similarity will fall here, that we will use MSMS similarity to compute subclasses, or we can also add the MSMS similarity modules here. But this tab, predict correlation modules, this is only using MS1 intensity data. It's not using any MSMS data or any other chemical or experimental information other than just MS1 intensity data. So with that, I could conclude this seminar that ChemRICH is an I would say is a novel and essential tool in metabolomics, untargeted metabolomics data interpretation, where it finds chemical sets that are important and that are associated with the phenotype of interest. But now we have a leverage to use different type of set definitions. So we're not bound just by chemical class or pathway. We can also use different other ways to group peaks in untargeted data, and then use those set definitions to run the set analysis. And in a way, it provides to look at data in different angles. So more angles we have, more biological insights we'll get, and that basically fits into that how powerful untargeted assays are in discovering these new sets. So what I wanted to give a strong message that do not limit your analysis to just this box here, that just do pathway as set definitions. Go beyond and run the class level analysis, define your sets in different ways, or find subclasses or mass spectral similarity modules for your unknowns, or include correlation modules or mass spectral these similar spectral modules. This way you want to include all your peaks in a set analysis. I think that's my core message, that ChemRICH 2.0 includes uh, all the peaks, so we don't miss any biological insights if there is any in an untargeted metabolomics data set. So thank you so much, and if you have any questions, you can reach out to me on this live chat box and comment box for the video. I will try to reply, or you can send me email anytime. Thank you.